Hi, so this is the last lecture, lecture 4, applying machine learning to omics data. So this will be quite relevant for the coursework that is going to be set. I'm hoping after this lecture you'll have a sense of how different machine learning approaches can be applied to omics data and actually understand what the results mean in terms of the biology. So we're going to look at linear modelling for differential gene detection, clustering, heat maps, dimensionality reduction, PCA and MDS, classification, and I'm hoping also it will give you a sense of how to read research papers about applying machine learning to omics data. Oh, I'm hiding this, but I'm going to use simple machine learning methods really just to highlight how you interpret the results in terms of the biology. Hopefully your other modules that you're taking, both supervised and unsupervised modules, will provide more sophisticated techniques that you can apply to, say, the coursework data. So the first technique is going to be differential gene detection. The motivation for this is to detect genes which are expressed differently between two samples. For example, we've been looking at the acute lymphoid leukemia and acute myeloid leukemia study of Golub. So differential gene detection would detect genes which are being expressed differently between the lymphoid leukemia and the myeloid leukemia. So those genes which are overexpressed or upregulated in the lymphoid samples compared to the myeloid, or those which are upregulated, overexpressed in the myeloid compared to the lymphoid. It's one of the fundamental things that bioscience researchers want to know. They want to know what genes are upregulated in certain conditions, or which genes are downregulated. And it applies to other omics data as well, which proteins have a higher concentration in some samples compared to others, which metabolites have a higher concentration compared to some samples. So you might recall the MVA plot from lecture 2 when we talked about normalizing data. So what would be the problem detecting differential expressed genes in this raw data? So this is the MVA plot. This stands for minus. It's the log expression of one sample minus the log expression of the other sample. So it actually gives you sort of log fold change. How much times different is it? So a value of +1, because it's a log scale, that would be a twofold upregulation of that gene going from one sample to another, and -1 would be a twofold downregulation of that gene. The A is the average, so it's the average expression of that gene between the two samples. So each point represents one gene. So this is the change of the gene, say going from one sample to another, and this is what was the average log expression for that gene. So if we did differential gene detection just with this raw data, what would be the problem? So I think there'll be a question inserted at that point. So the key issue here is that there's one of the chips is reading values which are sort of biased. They're on average much lower than the other chip is reading. So you actually detect thousands of genes here that are differentially expressed, which are not really differentiate differentially expressed. It's all an artifact due to systematic bias that hasn't been normalized away. So we need to normalize the data before we do any downstream processing. What if we used mean medium centered normalization? Would this be okay for differential gene detection? So hopefully you've answered no, it wouldn't be okay. Why is that? Well, the medium normalization relies on the medium value of all of the genes, and then you shift all the genes up by that amount so that that medium value, it has zero difference. It relies on the average difference of all the genes being zero, that the sample, most of the genes in the samples will not change, and also the ones that, the small number of genes that do change, they'll change both up-regulated and down-regulated. So on average it'll be symmetric. So this is what that normalization relies on. But the problem here is that the systematic bias is intensity dependent. So depending on the average log expression, if your genes have low expression, then it would say they're actually downregulated after normalization. And this would just be an artifact. It would just be because there's this systematic bias which is sort of intensity dependent. If the genes have high sort of expression values, they'll also be seen to be downregulated, and the ones with an expression value around 7 will be, on average, they will show that these are upregulated once you do mean normalization. So these systematically wrong sort of genes that are being detected due to systematic biases will hide genes which have a real biological upregulation and downregulation. So you might get 50 genes here and 50 genes here and I don't know, 100 genes all being upregulated, and there may only be sort of 20 genes that are truly changing. So all of this, these false positives will hide the real signal. So you'll recall that the way that this type of bias is handled is low-S normalization. We fit a locally smoothed line to it, and then in Windows we scale up all of the genes in that, essentially that window, to have on average zero difference, and we get rid of the systematic bias, which is intensity dependent. So after this global correction, we should be able to detect real genes which are really upregulated and downregulated by using the fact that this is probably the noise level, so this is the variation that you should use, and then some genes like this one has a value of 1.5, so it's potentially truly upregulated there compared to the background. So what measures do we use for detecting if genes are differentially expressed? Well, there are two orthogonal aspects. There's the size of the effect, and there's also whether the effect is statistically significant. So starting with the size of effect, log fold change is usually what is used in omics data to give a sense of the size of a change. So this is the log fault change plus 1. This essentially means that there's a twofold up regulation going from AML, which has an expression level of 1024, to ALL with an expression value of 2048. So it's been upregulated 2 times, so the expression ratio is 2, and then the log fault change, which is the log base 2 of the expression ratio, is plus 1. Similarly, if it's a twofold downregulation, it's minus 1. 0 if it remains unchanged, it's got a ratio of 1. The expression levels are equal, etc. So it's used to get sort of familiar with some of the terminology, and the terminology often gets mixed up in papers. So we have expressions, so this might be given by our red to green ratio for spotted microarrays, where the green is a reference gene set. So the expression is essentially intensity of the spot, and it gives a sense of how many mRNAs are actually bound, because that intensity relates to how many messenger RNAs are bound, or fragments of messenger RNA, with each one providing a fluorescent probe to increase the intensity. This scale is more naturally dealt with in terms of logarithms of it. So we get log expression, and for microarrays and omics we use base 2 to give a natural range for the bioscience. So 2048, the log expression is 11, because 2 to the 11 is 2048. This one's 1024, so that's 2 to the 10, so the log expression is 10. And then the ratio is, expression ratio is the ratio between these two, so it's 2 in this case, or this is sometimes called fold ratio. And then the log fold change is the log 2 of that ratio, so it's plus 1. But we can also get it from one log expression, 11, minus the other log expression, 10. It gives you plus 1. So this is why the MVA plot, it uses log expressions. It does one sample minus the other sample to get the log fold change. There's in research papers, and you'll notice I also do this, that often you call the log expressions, you sometimes call them just expressions, because generally all microarray omics data is done in log expression units. So you quite often just start referring to this as expression. The expression level of this gene is 11, whereas it's generally always a log expression value. So the terminology can become a bit confusing at times, what exactly is being referred to. But the log fold change provides a symmetric scale, unlike the ratios, and it gives you the size of effect. Most genes will only be upregulated to about +4 or +5, or -4 or -5. So we're talking a 16-fold increase or a 32-fold increase. So that's the size of effect. And then the other sort of key information is the effect statistically significant? So this is a sort of very schematic diagram. You clearly have so a t-test, in this case, if you assume it's Gaussian distributed data, a t-test would give you a p-value. But this is the general idea, is that we may have a twofold increase or a twofold decrease in both of these genes, gene 1 and gene 2. But if gene 1 says something to do with glucose metabolism, and the patient's diet is not controlled during the experimental setting, then the glucose metabolism enzyme may be all over the place from sample to sample. So this increase of this gene into experimental conditions, it may not be significant because of the larger variance in the actual gene itself, where some genes will be under very detailed control, and from one population you'll get a very narrow range of values. So this is a log2 expression value of 10, and this is around a log2 expression value of 11. So it's a twofold, a +1 log-fold change here, but this is actually probably significant because there's very small standard deviations within each of the sample populations. So this depends entirely on the gene. Each gene will have a different level of variance in the experiment. So yeah, you know sort of classic t-tests, and you've got sort of arrays. You may have the disease samples, you may have 3 of them. You have normal samples, you have 2 of them, so it doesn't need to be symmetric. And you can use a classic t-test to look at the average value of for each gene. Look at the average value of the disease samples minus the average value of the normal samples, and then working out the standard deviation is a matter of so this is a pooled, this is a sort of Walsh t-test. It does a pooled calculation for the standard deviation. So SP is calculated, so the variance of the pooled variance is the variance of the first samples, the disease samples, and SY is the variance of the normal samples, and then you have to take into account the weighting of each. So N is the number of disease samples, M is the number of normal samples, and this works out a pooled standard deviation, SP, and then the test requires the square root of 1 over n, number of disease samples, plus 1 over m, the number of normal samples. So you get a t-value, and then looking it up in a table of sort of t-test table with the correct number of degrees of freedom, which is n plus m minus 2, then gives you a p-value for that. Yeah, so 0.1, 0.05, so that's the typical threshold that you use, 5%, and these are the values of the t that need to be exceeded to meet that level of statistical significance. So that's one of the reasons that we always tend to use log ratios or log expressions, is that this turns potentially skewed data into something which is more Gaussian-like, and this is one of the underlying assumptions for applying statistical tests like the t-test and other methods, other parametric methods. So that's why we typically always work in log expression space. So there are a number of issues with this type of hypothesis testing of many genes, and you're probably quite familiar with these. But issue number one, multiple hypothesis testing. So if we have got a gene which is significantly differentially expressed at set alpha, or the p threshold of 0.05 or 5%, then what we're saying is that there's a 5% risk that we might detect this gene even though it isn't differentially expressed. It's the null hypothesis. So we take this 5% risk. Now this is okay if we have just one experiment. We know that there's a 1 in 20 chance that we will be wrong with this conclusion. But the problem from omics data and all other types of high throughput data is that we're actually testing 25,000 or 30,000 genes at the same time. So if we allow a 5% chance of each gene sort of passing the criteria, then this will immediately give, well, 1/20th of this number will be randomly differentially expressed. So we actually get 1,500 false positives. That can be seen as a problem if we only have maybe 100 differentially expressed genes, so by 2 differentially expressed, then this will be swamped with false positives. So this is one of the key issues with any high throughput method, this multiple hypothesis testing. One of the classic ways of avoiding it is Bonferroni method, controlling the family-wise error rate. So instead of having a threshold alpha, say 5%, the Bonferroni method adjusts the threshold so it is alpha over n, where n is the number of hypotheses being tested. So in this case it would be 5% over 30,000. Then we have essentially the chance for detecting one false positive amongst all of the data is only 5%. So it's a family-wise error rate. So that's very good. You've only got a 5% chance of actually having one false positive detected. But the issue really is that it's just too conservative. Generally when you apply Bonferroni correction, you just don't detect anything. This would be 0.05 divided by 30,000. It would be a p-value or alpha threshold, which is just tiny. So typically what we do use in omics data is false discovery rate. This uses a statistical model to work out essentially what is the error rate in the list of genes that are differentially expressed. So if we've got 100 genes which are differentially expressed and we have false discovery rates of 5%, then we would expect that 5 of them will be false positives. So we know that on average 95 will be genuine differentially expressed genes. This actually calculates a false discovery rate on a gene by gene basis when you apply it. But to get an idea of how essentially it's working, if you use a p-value of 0.0001, where we've got 30,000 genes being compared, we know that this p-value will result in, on average, 3 false positive genes detected. So if we use that and we get 100 genes detected, we know that there are 3 false positives amongst them on average. So we have a false discovery rate of about 3% within that data set. So this is it's essentially a modified p-value to make it slightly more conservative. So you might find that in a p-value might be 10 to the minus 6, whereas if you actually do a false discovery rate and you come up with modified p-values, they might go from 10 to the minus 6 to 10 to the minus 3 or something, depending on the data. So that's the first issue. A second issue is the small number of biological replicates. So when we use t-test, we need to identify, we need to determine what the standard error is, what the denominator is, the standard deviation. We saw it was a pooled standard deviation, and then the 1 over root n plus 1 over root m was really turning it into a standard error. It was, so typically if it's a balanced data set of n samples, you have standard deviation over root n is your standard error. So that's using that standard error to see how many standard errors are between one average x and the other average y of the two different samples. Now this sort of breaks down when we have the standard errors calculated from a very small number of samples, say 3 to 5, because then the actual error on the standard error can be very large, and every so often that standard error may become very small by statistical chance due to the small number of samples. So whenever, if the standard error is really, say it's got a value of 4, then it'll have a distribution around that value, and when it randomly becomes, say, 0.4 or 0.1, you'll start getting significant differential expression even when there isn't any. So we'll get a large number of false positives will arise due to having a standard error which actually has large errors in itself because of a small number of samples it's been determined from. So there are a number of ways of what's called variance stabilization. You want your variance to not sort of become small unless it really is small. So one of the approaches is actually to borrow information from other genes. And the classic sort of Smith 2004 approach to doing this, sort of shown graphically, is to, you've got your gene with its standard deviation, standard error calculated from maybe 3 samples or 6 samples, and to variance stabilize it, what you do is you choose other similar genes, and you can do this by having a window and choose other similar genes which have similar average gene expression values, and then you incorporate that information into your real standard deviation for that gene. So it's a sort of, you're including some aspect of the population, and the reasoning is that you hope the variance of the genes will depend on the average value. So they're sort of high, and then here there's a sort of tail pattern, a sort of fish shape, and that's what you're doing. You're borrowing you're borrowing information about other similar genes to stabilize the value, because you're now using many more values to calculate the variance. The problem with doing that is by stabilizing the value, you actually introduce a bias. I mean, these other genes here are not the same as the gene that you're investigating. So you introduce a bias. You're assuming these other genes will have similar variances, but they may not. As I indicated, each gene's unique. If it's a glucose metabolism gene, it may have a higher variance than, say, a cell cytoskeletal sort of housekeeping gene that's used to constantly build new parts of the cell. Then yes, these genes may not all have the same variance as the gene that you wish to calculate it for, but you're hoping on average that they do. So this is usually done in the Bayesian framework, where there's a certain amount of information from the gene itself, depending on how many samples you have, how reliable it is, and then there's a certain amount of information coming from other genes. So you find out, so you can see that there are considerations for omics data that you don't typically need to worry about in other fields. But your final result is a table of significantly differentially expressed genes, with acute myeloid leukemia against acute lymphoid leukemia. And the GOLOB data, you get this type of table. So we get a log fold change, so it's about -5, so that's actually a downregulation of 32 times, 2 to the -5. It's got a p-value of 3 times 10 to the -8, so it's highly significant. And this is the gene symbol, and this is a description of the gene. And you can see that cystatin C, cystatin A, so this gives you further support that the cystatin gene is downregulated, and because you've got, these are essentially independent sort of genes, they're isoforms, but they're both downregulated, which gives you further support that it really is downregulated. In terms of cancer, CDC25A cell division cycle is downregulated, so that's cancer related. And then you've also got genes, this is +2 about, so it's a 4-fold upregulation. It's significant, 4 times 10 to the -6. MAC-B is a myeloblastosis viral oncogene, so that immediately tells you it's myeloblastosis, so it's involved in myeloid leukemia, and it's an oncogene, so it's a cancer-inducing gene when it's mutated. So this is the final output, and a bioscience researcher would go down this list looking at what are the functions of these and how they might be involved in the disease under study, for example. We can turn all of these into a volcano plot to actually visualize them easier. So this is on the x-axis, the log fold change. So we've got a +2 is 4-fold up regulated, -2 is 4-fold down-regulated, -4 is 16-fold down-regulated. So this gene here is about 16-fold down-regulated in one of the samples. And then this y-axis is the -log10 p-value. So log 10, because it's natural, you talk about p-values of 10 to the -8. So this then would become -8 when the log 10, and then you do it in minus, so it's +8. So this gene here is 10 to the -8 is a p-value, and 16-fold down-regulated, so it's highly significant. So these are the interesting genes in this plot. So normally we have something a bit more complex than just a pairwise comparison, and I'm just going to touch upon this, but spotted arrays are often given as a reference design, or it may be a loop design where you don't actually have the comparisons you wish to make explicitly. There's often lots of different types of replication. There may be biological replication, which is the most useful, but there may be also technical replicates where you put the same samples onto the spotted microarrays or onto Affymatrix. You may have pooled samples, as we saw in one of the data representation examples. There were pooled liver, pooled brain, and I think pooled kidney samples. So that's where you take multiple samples, and because maybe you don't have enough sample from one person, you actually pool them together into one sample. So all of these adds complexity to the experimental design. And usually there are lots of other factors. There may be drug present, absent, stage of disease, male, female, may also be continuous variables, age of patient. So this is all complexities that need to be taken into account if you want to do differential gene detection. In this scenario, you've got confounding variables. And then we also have the multiple testing issues associated with 20,000 genes being tested and the small number of samples. So linear modeling is often sort of used in this scenario. This is in Bioconductor implemented as a package limmer. Extremely flexible, can handle all sorts of these aspects. I've put on a background interest paper on the Moodle site to show how limmer works, but it can handle single channel Affymatrix chips, so it can handle 2-channel spotted arrays with maybe sample A compared to sample B, or it may be a dye swap experiment, sample A compared to sample B, where this is red and this is green, and then you swap them, sample B compared to sample A, where this is red and this is green, to get rid of dye bias. But this is a slightly more complex design to model. And you might have a reference design. One of the samples has a dye swap, the other one doesn't. Or you might have this loop design. We have 3 samples and they're all pairwise compared to each other. So that's got a slightly different statistical design to analyze. And there's all of the aspects of different types of replicated samples and discrete factors and also continuous variables. So limmer can handle all of these, and it's got a sort of classic linear modeling. It's got a design matrix that takes all these factors into account, and then there's a comparison matrix where you have the comparisons that you wish to make, and it's sort of explained in more detail in the paper. I've also put down a further interest sort of workshop lecture on using bioconductor for this, together with data scripts data folder. So this is the App Studio, and it allows you to go through analysis of Huntington's disease data sets using AFI package, Limma package, and ANAFI, which is the annotation package. And you can get all of the genes and the gene symbols, the log fold change, average expression, t-value, p-value, and then this adjusted p-value. And you can see how it changes. You've got a p-value of 2 times 10 to the minus 20. Once it's adjusted for the multiple testing, 1 times 10 to the minus 15. And then the b-value is the log likelihood of that actually being a differentially expressed gene. So you can see these all highly significant. And you've got the genes here being highlighted on a volcano plot. So this volcano plot is slightly different. It's log fold change again, but because Limma uses a statistical model, it actually has the log odds on the y-axis, but it's playing the same role as the minus log 10 p-value. Okay, I think that's probably a good place to stop with section 1. The differential expression section is quite long. Even though the concept is very easy or simple, you just want to detect what genes are changed. The actual omics data, there are lots of aspects that need to be considered, and we went through some of them in this lecture. Okay, so section 2 is going to be about hierarchical clustering. Okay, I'll see you then. Bye.